Good morning. Ebba Ansen is a master student of mine, and she will be defending her thesis next Tuesday. You are welcome to Uppsala. Here I report only some parts of the work that she has done. Lars-Erik Liljedahl is Professor Emeritus of Quantitative Genetics in our department. He has been working with the question of aging since 60s. During these 50 years, he has shifted his views many times and has concentrated on different subjects. Recently, he has been concentrating on the question of heterozygosity and homozygosity as the age goes up. So the outline of the presentation is lots of slides for introduction, one slide for data, and then lots or few slides for the result. I do not have any slide for discussion. One slide for conclusion and one slide for acknowledgement. I hope this has been a very informative slide. Now, Lars-Erik, a couple of years ago, came to the department and met with Erling Strandberg, and he had some ideas. Erling sent these notes to me, and my reaction was that, well, this can't be true. This is too simple. This is too good to be true. And I was very skeptical about it, but Lars-Erik insisted, and we started to look more and more into the details. He has a long argument. It is a simple argument, but a long one. Step one is experimental populations, Drosophila, mice, chickens. These are the things that Lars-Erik has looked at, but we can look at other studies as well, that during aging, there is a higher expression of heterosis or a larger amount of inbreeding depression, especially for fitness-related traits. I will not go into showing the evidence. The references are in the paper. Now, heterosis or inbreeding depression can arise when there is a dominance-recessive relationship between the two alleles in one locus, or when we say the animal is heterozygote. However, this is a paradox. The reason is the animal genotype has been fixed at birth. The animal is not changing the genotype. But if the inbreeding depression goes up or heterosis goes up, it means that there are more heterozygote expressed. And this is, by definition, an epigenetic mechanism. The genes being activated or unactivated in the beginning of life, for example, more homozygote being active, and late in life, more heterozygotes. This cannot be unless there is an epigenetic effect. So this was the step forward, that one way to solve this paradox or dilemma is to accept epigenetic mechanism. Now the question is, what is it that we need to test this hypothesis? We need a measure, say, for example, lactation, milk yield for lactation 1, 2, 3, 4, 5, 10. And then we need allele frequencies, genotype frequencies for all SNPs that we can get our hands on, and probably direct information on methylation or acetylation on these different SNPs, and SNP effect estimated. So the first one we do not have. The second one, yes, we have. The third and the fourth one we do not have. So the message to Lars-Erik was, sorry, we don't have the data. However, something else can be brought into this chain of argument, and that is it possible that homozygous loci are more resource demanding? It might be the case, and it has been shown in both experiment and observations, that the homozygote have a higher turnover of the proteins. They demand more. Then, if this is the case, any SNP with a large effect, any gene with a large effect must be more resource demanding. And therefore, for the body, for the organism to carry on with this one, it must be the case that these large effect SNPs must have a lower degree of homozygosity. In that case, we don't really need a trait that has been measured during the life. Any trait would do, and then allele and genotype frequencies are available. Direct information on epigenetic effects are not necessary anymore, and when you talk about the SNP effect for different traits, you name it, we have it. So the message to Lars Erik was, we are back in the business. The only thing we needed was a stupid student to come up and take up this idea, and Ebba was willing to do so. And she did it in a very good way. So the data that we have is the intergenomic data. These are the data that has been, or result that has been routinely produced and distributed. Allele and genotype frequencies, 150 different traits are available, and SNP effects for more than 44,000 SNPs. So I'm just showing here that to show that the model has been sensitive, we have been having some result. This is for direct longevity or aging. And as you will see on top figure, we will have a SNP with large effect. It is present in some of the populations, but not present in the other ones. So we can see there is some variation. Now, what we looked at was actually, okay, we look at the SNPs that have different effects. We divided them into those that have effect that is half a standard deviation larger than average, 1.5, until 5 standard deviation larger. And as you see, these bars are showing you the absolute effect of the SNPs. And the lines are showing the degree of homozygosity. And as the argument that we put forward had predicted, these homozygosity degrees are going down as the SNP effects are coming up. And this pattern here is rather beautiful, but when you go to some other traits, you see when we come to 3, 4, 5 standard deviation away from mean, the homozygosity goes up. So there is something happening there as well. And the fact that homozygosity is going down, it doesn't mean that there is aging effect or some epigenetic effect. It might be just a case that allele frequency have changed. So let's look at these numbers that we see when we go up. When we come to 5 standard deviation larger than average, on average for here we are showing the result only on the Swiss scale. For these 31 traits that Switzerland has, on average there are only 10 SNPs that have such a large deviation. And these are the ones that are showing a lot of allele frequency change, not the other ones. Now, there is a trait group that you might have something that is measured during the lifetime. In fertility, the heifer fertility, and then it comes coming back to recycle until calving interval or days open. These are not necessarily separated from each other during the ontogeny. However, with some overlap, they might show a little bit of the aging process. And the only country that had all of these effects was Germany. And when we look at it, we see there is a strong reduction of homozygosity as the average effect goes up, except in the end. Now, as I was showing before, there is almost no change of allele frequency for these ones, but when we come to 3 standard deviations, they will start. And as you look at the direction, it might be the case that some of these alleles have a negative effect or positive effect, so their frequency is going up and down. Now, how much of it is because of allele frequency change, and especially if the allele frequency change is not at about 0.5, then selection is going to change them. Then there is obvious predictable changes of heterozygosity, homozygosity. But here we looked at the panmictic index, and you expect it, if everything is normal, the panmictic index to be around 1. This is the observed number of heterozygosity divided by the expected number. But as you see, we have a very low value, especially for the ones that have very small effect, and there is no frequency change. So I don't know how to interpret this result. Why do we have low panmictic index? It might be the case that there is a bias in the AB allele coding that Illumina. As you know, that the B allele has a frequency of 54% versus 46 for the A allele. Some of it might be the case. So the conclusion is that the byproduct of a routine national genetic evaluation, genomic evaluation, or international genomic evaluation can be used in many other areas of research with not much effort. Then the analysis that we have done here was a very simple one. We just used, we could use Excel, even though we used SAS. And we have enough evidence up to now, looking at this data, that there actually might be some epigenetic effect affecting the expression of homozygote, heterozygote, and it warrants more emphasis or more investment on this subject if we get funding or we get interested PhD students. I would like to acknowledge all intergenomic partners that we had access to the data and being allowed to use the data from them. Thank you.